Hello everyone, welcome to a new video of Nucleus and Mishael Wright, and today we're going to introduce the fascinating world of cells. We're going to approach different dynamics. First of all, we're going to talk about the history of the cell, how we came with this amazing concept, how it formed, and at the same time, we're going to see how were conformed its organelles. Secondly, we're going to talk about what are the differences between eukaryotic and prokaryotic cells. And finally, their functions, structural and functional speaking. Okay, 4.3 billion years ago, we had extreme conditions in what we currently know as our planet. So we had something that was called the Hadean Eon. Hadean Eon is related to the Greek mythology god that is called Hades. Hades was a great god of the underworld as we currently know it. So after that, the Earth planet cooled down. So we have high temperatures, we have a really hard conditions for what we know as living beings. And the cells were something that was an impossible idea to be done, right? So the Earth planet cooled down, okay? Under these hard conditions, we had a lot of volcanoes. They presented, as the name says, a volcanic activity. So this volcanic activity started delivering some gases. Some of them were ammonia, methane, right? CH4, sorry, ammonia, NH3, carbon dioxide, and nitrogen. Okay, so really important to comprehend that these two that are here, methane and carbon dioxide, are the famous greenhouse gases. Okay, related to the greenhouse gas effect. So on the early Earth planet, we had a lot of volcanoes. So the volcanic activity was tremendously high. Then we had the Earth planet under temperature average conditions of 400 Celsius degrees. And we had some distributions, right? So the distributions were 95% of carbon dioxide. We had 4% of water vapor. And then we had the other traces of nitrogen, ammonia, and methane. Okay. So in the current conditions, your planet is mostly covered by water. More than 74% of your planet's surface is covered with water. Ironically, we call this your planet, but it should be called the water planet. Okay. So the distribution of the percentage is going to be different by the way. So here we will have a distribution of okay 78% is going to be covered by nitrogen, 21% is going to be covered by oxygen, right? And the other distribution are traces of water vapor, methane, and ammonia. So you can find out a great difference in contrast of the initial conditions of the Hadean era to these current conditions. Okay. Right, the interaction between these ultraviolet radiation and in these temperatures and conditions are being created different compounds of carbon. So if everything had been added to the gases, possible building blocks of life could have been created, such as nucleotides, oligo, oligo, nucleotides, peptides, ribose, pentose, fatty acids, amino acids, so 3, 4, 5, 6, 7. Okay, so this idea came in 1924 by a scientist whose name is Alexander Oparin, right? Who led us to a concept that we know currently as the primordial soup. Okay, so he basically talked about something that we know as the co-surveys. And it's where these prime molecules, big molecules that contain a repeating structure, and they were like the pre-existing structural form of cells. So really found being redundant. Okay, so before the cell, Alexander Oparin proposed that we had conditions that led us to be in a primordial soup, where these molecules used to consume other molecules, right? The coagulates, and then posteriorly, in the Earth planet, we had a big structure of these coagulates because they have consumed each other, looking for energy. Okay. And after this, we had the chemical processes that divided all these soup and that gave us all the cellular structures. Right. However, in 1929, J.B.S. Haldane sharpened his theory. Right. It has been demonstrated that high energy levels in radiation can break down molecules. An example of this is the famous pyruvic acid. That is a molecule that has certain incidences in metabolic pathways of cells, right? So, so what does happen with it when this molecule is exposed to energy and light conditions? It breaks down and transforms into smaller molecules that will undergo different chemical reactions. And these micromolecules will have a high reactivity. All right. So after this, some researchers have shown that certain molecules like nucleotides in these bases, they have kind of protection. So the basis of nucleotides, right? Bases of nucleotides bases protects. This base protects the pentose sugar, and also the the phosphate groups, right? Presented in a famous molecule that we know as the RNA. So, in certain way, we could have seen that the ultraviolet radiation could be a limitation, but maybe it was a natural selection processes that took place. And you know that from the RNA, we can have something that is called the protein synthesis. So, according to this, we can say that the RNA started forming larger and larger molecules. So, here is where we have something that is called the central dogma of molecular biology, right? The molecular biology central dogma, where we state that this simple and simpler change of RNA, simpler than those ones that appear in the DNA, we can have and obtain amino acids that are the building blocks of life. And amino acids will considerably give us or give rise or yes, give origin to proteins. And those proteins will have different functions. They can be structural and at the same time they can be functional. Now we're going to talk about the cell as the smallest unit of the self-sustaining life. As far as you know, the cell is a functional and structural unit for all living organisms. All cells will contain a plasma membrane that will protect the nucleus, which is an organelle of it. The nucleus contains the DNA that is used as a template for the RNA and the protein synthesis. In the definition of life, we cannot just approach the functions of the cells, which are their relation, the respiration, you know, reproduction, and so on. But we have to use three principles to define life in and of itself. The first principle talks about that living organisms must be separated from its environment, and this will allow us to have internal processes. The second principle states that the organisms will have the functionality and option to reproduce themselves. And the third principle is more likely to approach living organisms as chemical and physical machineries that will perform processes inside themselves. NASA company has worked on these definitions in a more profound view. They use Darwinism and they state that that all the living organisms will have this Darwinism point of view, which is that they will have to evolve. And to evolve, will have to carry out other functions, which are the natural selection process, adaptation process, and the reproduction. But we have a great problem in this definition because there is a great or huge limitation on it. And that limitation is about that we have discovered life, but only on the Earth planet. So, but I would like to invite you to wonder about this. Okay, currently there are certain studies that will take the human being to research on the Mars atmosphere. There are like a certain bodies of area that will contain sub layers of water. Okay, maybe there could be life there, but we don't know those conditions in a profound view. So those are the limitations on the NASA on the NASA's definition about living organisms. Okay, on the other side we have viruses. Viruses are not classified as living organisms because they don't share some characteristics with cells. And one of them is that viruses cannot evolve. Viruses also don't carry or don't perform the homeostatic process that allows us to regulate the temperature according to the external conditions, creating kind of balance between the internal and the external conditions. Okay, but they can replicate their genetic information in forms of DNA and RNA by using the protein synthesis. On the other side, we're talking about Mars. Mars is an excellent and promising place because there are some sub pockets of water that will have a promising option to be living life out there on this planet. Cell spontaneous generation. So there is a big question that we all wondered. And this question was about how did these first cells evolve? Okay, the answer can be if we look through their internal processes. So we have to characterize three conditions that take place in cells. Those three conditions are that cells are contained in with a plasma membrane, and they are able to take their hereditary material, which is the DNA, hereditary material, to the other cells. Okay, on the other side, we can see that they respond to the external stimuli. Okay, something that does not happen in viruses. Okay, not in all the cases, but it generally doesn't happen. And also that they have internal chemical and physical processes inside themselves that will allow them to take other big and macro processes such as the growth, such as the relation, such as the respiration with the adenosine triphosphate, which is ATP. However, we gotta talk about the spontaneous origin of cells. And maybe the answer can be out there. Maybe the cells came from non-living components or compounds. Okay, for this, many scientists have wondered about the first process of evolution in the first cells. Okay, so to take this into consideration, we are going to enclose three important processes that are given in cells. This is the plasma membrane complement. Okay, so we can define the cell as something that can be enclosed by a plasma membrane that at the same time is able to interact with its surroundings, in a complement, in a continuous interaction with internal and external conditions. Okay. Also, you can say that there is a great importance in the genetic or hereditary material. Okay, so all the cells can transport their material to other cells by inheritance. The other one is going to be that they are able to perform functions that require energy, right? That will be taken to go through more processes that are going to be called macro processes. Okay, so this means that they will be able to take internal and using their chemical and physical processes to perform processes such as respiration, growth, and other ones, right? So this has taken us to define life. Okay, but at the same time, we have all the concept that is evolution. We have been taught by some scientists like Lamarck or Darwin that all the organisms evolve. In today's evolution, we gotta go through natural selection under pressure of certain conditions that is called environmental pressure. We have to go through all the processes such as adaptation to the environment, and this adaptation is closely related to mutations. Okay, so there are different types of mutations. We have mutations, okay, and we're going to divide mutations into three different categories. So the first is negative, neutral, and positive, or better known as beneficial mutations. Okay, so this happens when you have different microprocesses such as an insertion of a nitrogenous base, you have a deletion of a nitrogenous base, you have a replacement of a nitrogenous base, when you have a network, right? Okay, and so on. Okay, we're gonna see different microorganisms, and remember that the organisms can be classified as unicellular, pluricellular, or multicellular organisms. And in some cases, organisms of complex cells, because there are organisms that just consist of 7 cells, 8 cells, it depends. Okay, but today we're going to see the unicellular ones. And this is the chlamydomonas. Okay, the chlamydomonas is kind of alga. It's an alga that is in habitats rich in ammonium ions. Okay. All right, so the structures you can observe here are going to be the flagella. This one that is in here, flagella. And this is the light sensitive spot. This is the nucleus. Okay. This is a famous, okay, I'm gonna write here, famous cytoplasm, or in some cases known as cytosol. Okay. This is gonna be the starch storage. And finally, we're going to have the 6 parts: 1, 2, 3, 4, 5, and 6, which is the chloroplast. Chloroplast. Okay, so then we have another protozoan.
What is the length of that?
All right, the length of this is 30 microns. Okay, 30 micrometers. And okay, so thank you. So let us go with the other one, that is the protozoan. Okay. That is in fresh water. You can find it in fresh water ecosystems, environments. And this is the famous amoeba. The amoeba will have some structures, and they are these protuberances you can observe here that are known, as the pseudopodia. Pseudopodia. Okay, they will allow these organisms to move. It will allow the movement in these organisms. Okay. Then we'll have the vacuoles. There are these ones. Vacuoles. Okay. There is going to be two types. There's going to be two types of plasms, right? So we have the endoplasm, endoplasmic structure, and we have the ectoplasm, the ectoplasmic structure. Okay. Okay. Then we'll have the nucleus, which is this one, nucleus, and this is the contractile directly backward. Then we're gonna go into another famous, right? So we had seen these multiple microorganisms in fresh water, rich in ammonium ions, which is the plankton. We have the amoeba that is also in fresh water, and this one can be found in your digestive system. Yes, the length of this amoeba is 400 microns. Then we have this other one that is known as Escherichia coli. She's a bacteria. She's a bacteria. So we'll have the peeling, right? Then we have the plasmids. Plasmid. Here, these points will be the ribosomes, okay? And we'll have our genetic information in a circular form, and this is the famous circular DNA. And we have the cell wall, and the cell wall will be formed by oligosaccharides and amino acids. So the value of this is going to be 2.0 microns. And these are the first organisms we're talking about. Because according to the cell theory, you can say that, okay, so unicellular organisms can carry out all of the functions that are taken by the cell. Okay? They will do it by themselves. Not, this is not as in our case, because in our case we have many cells working for us. So they can do this. They can do their cellular processes, but at the same time they will be classified as organisms. Okay, so now let's approach the cell theory. The cell theory states that cells come from pre-existing cells. The second one statement is that all living organisms are formed by cells. Okay? And the third one states that unicellular organisms perform the functions of cells. That cells can perform their functions only until one specific and determined time. Okay, so in the last part, we have seen that we can make some statements about cells. Cells are the foundation of life. Okay? There is a single living being without cells. They vary in size and function. So remember that microorganisms that are unicellular can carry out all the cell's functions. And the microscope helps us to understand and verify the cellular structure. Okay, so it's really crucial to comprehend the two great divisions that we have. We have unicellular organisms that are conformed by only one single cell. And in this kingdom, you can find in the prototista kingdom, you can find two types of organisms that we have previously seen. Uh, the Amidomonas and the amoeba. Okay, and the other one, this is supposed to be written differently, but I will leave it like that. Okay, and the other one can be bacteria. Okay, so we have the protists. There are these two ones, and an example of unicellular organism. Other can be bacteria. Okay. Okay. Then in the bacteria, you can find Escherichia coli. Escherichia coli. Okay. Okay. Then we have another classification that are multicellular organisms. And examples of these can be mammals, like you, like me, and fungi kingdoms. At the same time, you can find, for example, some flowering plants. Flowering plants. Okay, so to analyze cells, we'll have to talk about a great instrument that was implemented in the 19th century. Uh, and the instrument was called the microscope. Okay, this is one of the most modern microscopes. This is a light microscope, and we'll explain you its parts. So first, we're going to have the lenses. Okay, so here you place your your eyes, and these are called the eyepieces. Eyepieces, right? They normally are going to be 10x. Okay, so then we will have this is the frame, and the frame will contain a rigid structure that will give the microscope a balance. So we're talking about the frame, right? Now we're talking about these screws that are the coarse adjustments. Okay, so they will give us a general image of the sample that we're analyzing. But they're going to be called the coarse adjustments. Okay, so the technical name for this is going to be coarse adjustment. Okay, and okay, so there are some microscopes that have the four different objectives. So the idea is that you multiply the size image times the objective. Okay, so there is a multiplication, and you can draw like okay. I'm not keen on this kind of triangles to apply formula or equations, but you can say that the magnification equals in the size of the image times the objective. So you can obtain a, a magnification. So if you multiply, for example, 4x times the 10x, the magnification equals 40x. Okay, so this is what we apply. So you also can play between these formulas, right? You can obtain the size of the object just by dividing the magnification and the objective and so on. Okay, so there are we're not gonna be talking about that now. So let us continue. So here we have some clips, and these clips that you can observe here are going to be to hold the sample, right? These are placed on what we call the stage. Okay, this is the stage, so the clips will be placed here, and you can play with them just by taking and assuring your sample. So here we have the tissue or skin you can apply You can apply some substances such as the local solution. You can apply some tinctures, some tinctures like the Wright ink to observe some organisms or organelles of the cells, such as those ones that contain almidon. And okay, among those organisms, we can find the nucleus now, or some proteins using the different substances. So we have, for example, the local solution. Local solution, okay. Okay, there is another one that is the right tension. So you can analyze some organisms depending on the solution you're using. Okay, so then we're going to have a debate. This is going to be a connection led by the frame. So this okay, you can take the microscope, you can hold the microscope just by taking it by its frame, and it will be connected to the base. Okay, so there we'll have the mirror. And the mirror is going to reflect. Okay, so here you can find the mirror, or you also can find some lamps, no? So there are some lamps that will be the source of light, or light source. And you can regulate that light that will be reflected towards the sample just by taking this strip. You can see that is called a condenser. And that condenser is a regulator that exists to allow the pass of light, or the amount of light you want to use in your sample. Okay. What else can I tell you? Okay, so this is basically the microscope, and the microscope was implemented in the 19th century. 19th century. Okay, and it was like a tiny and simple structure. So it wasn't that elaborated as nowadays, by a person whose name was Anton von, this is von Leeuwenhoek. Okay, so after this was sharpened by another scientist whose name was his name is Robert Hooke. Okay, so Robert Hooke was one of the fathers of the terminology of cells, because he first observed the cells in a cork that was found in a wine, and he observed some structures that in Spanish you can call cells, as if they were like these geometrical structures, right? That is why we call it this way. So the cell. Okay, and he discovered in plant cells that all the organisms had this tendency to be formed by cells. So remember the cell theory. Okay, so we have some experimental techniques. So we're going to take the preparation of a temporary mount. And remember, the cells can be mounted on slides so they can be viewed under a microscope. And this can be disposed of once the cells have seen and studied. Stains can be used and view cell features more clearly. Techniques describing how temporary mounts can be made are outlined here. So living cells are not only very small but also transparent. In light microscopy, it is common practice to add dyes or stains, as I have explained you before, to introduce sufficient contrast and so differentiate structures. Dyes and stains that are taken by cells are especially useful. Okay, so we can observe in the nucleus, the cytoplasm, and cell membrane in human cheek cells. And this is an experiment that I would like to show you. So here I'm showing you some photos about an experiment we performed with our students, taking samples of their cheek cells. Okay, so you can use, for example, a lining of your cheek using a fridge and use cotton, but you remove from the back. Thousand materials removed by cotton, but onto the center of the microscope slide and add a covered sleeve. Then dispose of the cotton bud safely and hygienically. under the microscope, slide yourself in at the end of the observation, immerse slide in 1% sodium hypochlorite solution to sterilize the slide in cold sleep. To observe the structure of human chick cells, you can irrigate the slide with a drop of methylene blue stain, following the procedure that I'm showing you right now. So this is like the mounted needle, the covered sleeve, the microscope slide, and chick cells here. So here we have an image of the irrigating temporary mount. Okay, so this is the figure 82.2.4, preparing living cells for light microscope. So you could also observe a chloroplast in most leaf cells or the nucleus, cell wall and vacuole in an ion epidermis cell. So these are the images that we're going to be approaching about the eyepiece reticule. The size of a cell can be measured under a microscope, transparent scale, called a reticule. Is mounted in the eyepiece at the focal plane. There is a ledge for it to rest on. In this position, when the object under observation is in focus, so too is scale. The size, for example, length or diameter of the object may then be recorded in arbitrary units. Next, the reticule scale is calibrated using stage micrometer. Remember that the microscope is going to be using some measures that are even smaller than millimeters, and they are going to be called microns or micrometers. So, in fact, a time transfer ruler, which is placed on the microscope stage in place of the slide, and then observed with the eyepiece and stage micrometer scale, superimpose the true dimensions of the object can be estimated in micrometers. So, show how this is done. Okay, so we're going to talk about magnification, and for magnification, we're going to use three variables, which are image. This is the actual size of image. This other one is going to be magnification. Okay, so from here we're going to obtain three different expressions. The first one is that image equals actual size of the image times magnification. This is going to be my first expression. The other one is going to be actual size of the image equals image over magnification, and magnification equals image over actual size of it. Okay, so there are different ways. In the scientific community, we don't like to use these kind of pyramids that are used in different expressions, for example, to obtain current, voltage, or resistance, right? Where you can obtain a voltage equals current times resistance and so on, so you play with them, right? But now we're applying this to the microscope. I would like to showcase you different ways to obtain these. Okay, so that is like the easiest way. But we have a number of different strategy we can implement, and it's just by taking that the actual size of the image times the magnification will give me the image in and of itself. And I can continue playing with this, right? So I can say that, for example, to obtain the actual size, so I will operate magnification in both sides to clear it out. So I would say that in this case it's multiplying. So I will need to divide it, right? So magnification times 1 over m, so I can cancel it out. And here I will do the same, right? So I would say I divide the image over magnification, and this gives me this. So actual size of the image is image over magnification. Okay, and this is going to be my first expression. Okay, the second one can be, for example, let's suppose I want to obtain magnification. So I have this expression. Okay, and from this expression I obtain this other one, which is the actual size. But I want to obtain magnification. And what about this? So I would like to talk about magnification and how to get to this point in two different ways. So I will show you the shortest one and I will show you the longest one. And I will explain why it's better to use this way, but it doesn't mean that the other one is not important for you to understand the mathematical expressions or the mathematical procedure. So let's suppose we want to obtain a magnification. So I would say that the actual size of the image equals I over n, right? As you want to obtain magnification, this is divided. So in this case, as this is divided, I will have to clear the magnification just into steps. I would say magnification times actual size of the image equals image. But this is the expression that we had in the very first time. Okay, but what I want to look for is for the magnification. I want to clear it out. So in this case, I would say that magnification, so this is multiplying and I operate the actual size of the image in both sides. So as this is multiplying the other size, in the other side, okay, I will divide it to cancel out these two terms, and in the other side I will have to divide it at the same time. But I'm not gonna do that, right? In many ways we say, okay, so the expression goes to the other side of the equation or of the input just by with the opposite operation. But those are not technical terms because currently what we do is that we operate in both sides. So what we have to do is magnification equals. I operate it, but I'm not going to write that because it would be so long. I will write the other one, which is long, you know. So I would say image over the actual size of the image. So I have the other expression, right? So I have this one, which is the first one. I have this other one, second expression, and I'll have the third expression. So let us see if it fits with the things that we did first. Okay, so I equals 8 times 7, right? This is okay. This is okay. Okay. A equals I over n. This is okay. This is okay. And here m equals I over a. This is okay, and this is okay. But okay, so there are some questions that are raised on me, and those questions are okay. Let us take this part, the third expression, and what I want to find out is the magnification. But I don't want to do two steps. Let's call these two steps 3.1 and 3.2. Okay, let's call it this way. I don't want to apply these two steps because I consider that, okay, in a more linear way of thinking, I would say, okay, but this is simple. I want to obtain magnification, just let us play with the variable of I. Okay, so how would it be in this case? So so I have this expression. I want to find out magnification, which is the topic we're talking about today. So the actual size of the image equals image over magnification. So, okay, now I will try to apply different properties. So I will take the magnification to this side, okay, but I will clean it out. So what I will do would be something. I have actual size of the image equals and I want to delete the I, okay. I want to eliminate it from my expression. So I will apply I is divided, so I will have to say 1 over I, right? And as I am putting this expression here, I will have to place the same expression in the other side. So I would say 1 over I, okay? And okay, so here is where we cancel out these two terms. And finally, we will have that 1 times A equals 1 over I and equals 1 over N. 1, or sorry, this is 1. 1 over N. Okay, but I have something now, and that something is that I have a number 1 in my numerator, and I have the M in the denominator. So I want to place the M in the uppermost part of the fraction. So what can I do? Okay, there is a property. I will continue, right? So this is step number 1, step number 2, step number 3, and this is step number 4. Let us go about steps, okay? So I have the expression which is A over I equals a 1 over M. But I wanna place the M in the uppermost part. So I would say that A over I equals M, and according to the property of exponents and fractions, I can say M to the minus 1, or I can say M power minus 1. Okay, you can say this in both ways. Okay, so but I continue with the same problem because I have the exponent, and the exponent is negative in and of itself. So what I would do in this case is that in my fifth step, I would say that if A over I, okay, and I will apply a power of minus 1 in this expression because I need to convert the exponent into a positive 1. So I would say that M to the minus 1 times power minus 1. So as you can see, right, so there are like some properties such as, so when I say A to the minus 1, and that is power to another in our power, I would say that this equals to A minus 1 times minus 1, which at the end will give me positive 1, A plus 1, which would give me A, right? So applying this property here, I would say that so this is 6, step number 6. So I would say that this will become into A minus 1, okay, over I minus 1 equals M. Okay, but there is another problem here. And it is nice to complicate stuff because in that way we can understand the things in a deeper way. Okay, so what I would say is that in the step number 6, I would say that A equals 1 over A, 1 over A. I want you to focus on this, over 1 over I, okay, equals, and because this is an equal, okay. And finally, if you operate this strings times strings, you will obtain that I will write so M equals, so I, because 1 times I equals I over A. And this is the way that we got into the expression of the objective. Okay, so we say that M equals I over A, closely similar to this one. Okay, So that is why, okay, you can go directly, you can clear out this easily, but it takes a lot of time, but it's nice to do it because believe me, you have to do it to understand it better. There was a friend of mine who he had a more mathematical mind and he explained a lot on how to get into this equation. It's nice, right? So we're not forced to know everything, but it's nice to have critical mind and critical thinking and ask questions, formulate questions about how we should do this stuff. Okay, so now let's talk about what we're talking in this case, which is objective, right? This is the objective in this case is the magnification. Okay, so the magnification is an important expression that I would like we place on the table today. It's negative in and of itself. So what I would do in this case is that in my fifth step, I would say that if A over I, okay, and I will apply a power of -1 in this expression because I need to convert the exponent into a positive one. So I would say that m to the -1 times okay, power -1. So as you can see, right, so there are like some properties such as, so when I say A and to the -1, and that is power to another in our power, I would say that this equals to A -1 times -1, which at the end will give me +1, A +1, which would give me A, right? So applying this property here, I would say that so it's 6, step number 6. So I would say that this will become into A -1, okay, over I -1 equals M. Okay, but there is another problem here. And it is nice to complicate stuff because in that way we can understand the things in a deeper way. Okay, so what I would say is that in the step number 6, I would say that A equals 1 over A, 1 over A. I want you to focus on this, over 1 over I, okay, equals, and because this is an equal, okay. And finally, if you operate this strings times strings, you will obtain that I will write so M equals, so I, because 1 times I equals I over A. And this is the way that we got into the expression of the objective. Okay, so we say that M equals I over A, closely similar to this one. Okay, so that is why, okay, you can go directly, you can clear out this easily, but it takes a lot of time, but it's nice to do it because believe me, you have to do it to understand it better. There was a friend of mine who he had a more mathematical mind and he explained a lot on how to get into this equation. It's nice, right? So we're not forced to know everything, but it's nice to have critical mind and critical thinking and ask questions, formulate questions about how we should do this stuff. Okay, so now let's talk about what we're talking in this case, which is objective, right? This is the objective in this case is the magnification. Okay, so the magnification is an important expression that I would like we place on the table today. Okay, and we finally return to this, okay? But it's okay. It's like ironic. It's nice. So, as we have said before, magnification, I will take this expression, right? Magnification, I over A, right? So, magnification equals I, which is image over the actual size of the image. And let's talk about an example. So we have an example. In this example states that I need to obtain a magnification. I know that the image is going to be 15 centimeters, and that the actual size of the specimen is going to be 150. Okay. So, but there is a problem because when we talk about magnification, we have to use some units that are called microns, no? So the microns are units that we use and they are even smaller they are even smaller than those ones that are in the centimeters. So we say that one micron equals the okay, sorry. We're going to say that 1 centimeter equals 1,000 10,000 microns. Okay. So in this case, we need to convert the 15 centimeters into microns. How we do that? So we say 15 centimeters times the we need to cancel out centimeters. That's why I'm placing the denominator here, and I say 1,000 microns. Okay. So I just cancel out these two units, and I would say that this equals 150,000 microns. Okay, and then I'll take this and I will operate my image over the actual size of it. So I will say that this is going to be 150,000 microns. Okay, 150. I will cancel it out, and I will say that this equals to the and this equals to 1,000. Okay, so as we said, it's crucial to understand that as we're working with microns, we're working with a microscope, so we have to use microns here. So here you forgot to write it. And the magnification equals x 1,000. But okay, let us take another example, and I would like to throw at you before you to do it. So try to do this other one with different units. Okay. So here you got a magnification equals. So let's change the image. So let's suppose that in this case I'm not going to be working with an image of 15 centimeters, but I will use another image that will be 40 centimeters. Okay, this is microns. So I need to convert in the image of 40 centimeters first into microns. But there is an exercise that you can do, and is that you can also convert in microns into centimeters to operate similar units. Okay, as we're talking of microscopy, you better convert in centimeters into microns. So I would say that in microns, 40 centimeters, knowing that 1 centimeter equals 10,000 microns. Okay, so I would say that I cancel it out, and I would say that this equals to 400,000
microns. So then I take those 400,000 microns here. We say magnification equals the image, which is 400,000 microns over 150, and you can cancel out these two. So that you say that 40,000 over 50, and this would give me a result of magnification. This is with microns, sorry. All the time we have to use the units. Okay? So this is a magnification of 2,666 okay, x. This is periodic, I would say. Okay, so 66. So that would be the result in this case if I change the the size of the image. Okay, the image. And that's how we work with the microscopes currently. Okay, so we said that the magnification, sorry, equals image over the actual size. So let us suppose I will have to find out my magnification where the actual size is going to be 450 micrometers, and I have an image of 15 centimeters. So we convert the 15 centimeters into microns. So I would say that 1 centimeter is 10,000 microns. Okay, so I cancel it out. Now I would say that 15 centimeters equals 150,000 micrometers. Okay, so I'll use this and I will operate it here. So magnification equals 150,000 micrometers over 150 micrometers equals magnification of. So I can cancel this out easily and it will give me like x 1000. Okay, so I'm gonna use other example to show you, and let me write the other example here. Example number 2, where I will change the image. So I would say that the magnification equals image over the actual size, and the magnification will continue with the actual size, which is 150 micrometers, but I will change the image by 40 centimeters. So then I will have to change these centimeters into micrometers. But it's really important to highlight that you can change it. You can change the micrometers into centimeters, and you can obtain the same. So let us see. In this case, I will convert it here. Okay, so I would say 40 centimeters times 1 centimeter and 1,000 micrometers. So I cancel it out and I finally will obtain 400,000 micrometers. Okay. So then I will take this value and I will replace it here. just to say 400,000 micrometers over 150 micrometers, and this will give me 2,666,000 periodic, 2,066, 666 periodic 6 micrometers. Okay, so the magnification at the end is going to be 2006 2666.6. Okay, this unit is not micrometers. Okay, so I would like to place, right, to expose you another example. We'll continue working with this, but we are going to convert, in this case, the micrometers into centimeters. Okay, so before we're approaching this unit, working with micrometers, but now we're gonna approach the same problem but using centimeters. So we'll have to write the equation, and my image is going to be 15 centimeters over 150 micrometers. So as I had this in the previous example, I converted this into micrometers. What I will do will be so like converting the micrometers into centimeters. So I will take this singular rule, which says that 1 centimeter is going to be 10,000 micrometers. Okay, so how many centimeters are 1 micrometer? So settle down that we multiply 1 times 1 divided 10,000. So what I would do would be something like 1 centimeter times 1 micrometer, 1 centimeter micrometer, over, because this is a multiplication, this would be a division, over 10,000 micrometers. So we cancel out these two. And finally, I would have that this would give me 0.0001 centimeter. So I would say that 1 micrometer equals 0.0001 centimeter. Okay. Okay, so once I have this, I will multiply this times 150. So I will take 150 times 150, okay? So in a singular relationship, I could say that 1 centimeter equals 1 over 10,000, okay? So it's the same, and I can say that 150 is gonna be like 150. So that's gonna be 150 over 10,000. That will give me 50 over 1,000, and I will divide 50 over 1,000. That will give me 0.050. Okay, so that's gonna give me 0.015. So already centimeters. Okay, so then I'm gonna take these 0.015 centimeters, and I will operate these here. I will say that 15 centimeters over 0.015 centimeters, and I will cancel out these two units, and that will give me X magnification. So I obtained the same result, but from a different viewpoint using different units. In this case, we convert the micrometers into centimeters using the unit rule and operating it. So you can verify this with calculator. Okay, now we're going to talk about resolution. Resolution is the ability to separate relatively small objects. If you ask me what is more important comparing resolution versus magnification, I would tell you that resolution is more useful because you can have a great magnification, but if you have a good resolution, it's not going to be possible to see some structures. In this case, I would like to bring on the table something that is called the chloroplasts. And the chloroplasts are organelles that are going to be participating in the color of plants that is called chlorophyll, and at the same time it's going to be participating in the photosynthetic process. So the chloroplasts are going to have some internal and external structures, and those external structures are going to be called the external membrane. Okay, external membrane. So we only can observe the external membrane with a high resolution, but at the same time you can have something internally that is going to be the internal membrane, okay, internal membrane. So you can have, for example, a great magnification as I showed you in the example, but if you don't have a good resolution, or in other names it's going to be called resolving power, resolving power, resolving power. Okay, we'll not be able to observe the differences between these tiny organelles parts. On the other hand, I will tell you that we have different types of microscopes, and the types of microscopes can be classified. For example, one of them that we use a lot in the laboratory is the light microscope, and the light microscope has a limit in its resolution. The limit for light microscope is 0.2 micrometers. Okay, and these structures can be, for example, seen under another type of microscope, and that is called the electron microscope. Okay, capitalize this one. Okay, so the electron microscope will allow you to see smaller structures with a great resolution also. So it's really important to know that resolution works with the wavelengths of light. And remember that in some classes ago we saw the electromagnetic spectrum. Inside the electromagnetic spectrum, that is going to be like a scale. I'm just going to mention three different types of waves you can find there, right? So its wavelength will be reducing, right? So we're going to have the infrared waves, such as, for example, it's going to be 10 to the minus 5. Okay, so the wavelength for infrared waves is 10 to the minus 5. But we human beings can see by using the visible light. Okay, so the visible light is going to have a wavelength of 0.5 times 10 to the minus 6. Okay, and that's going to be located here. Okay, so also after this type of wave, we will have another ones that will be even more reduced. We will have a shorter wavelength, and their name is going to be called ultraviolet waves. Okay, and you can find them in some hospitals, for example, to analyze patients. But I'm not going to go into details here. So that's going to have a wavelength of 10 to the minus 8. Okay, so the resolution works with this visible light. So the visible light will have different types of colors. There are Roy, G, Yeah. Okay, where we have red colors, orange colors, yellow colors, green colors, indigo, blue, and violet colors. Okay, so the closer we get into this part, that is another type of wave, the less wavelength we will obtain. So the distance between crust to crust or trough to trough is classified as or is named as a wavelength. And what I want to tell you, so resolution works with wavelength. And wavelength, okay, the visible light will have, generally speaking, will have a great wavelength. So what resolution does is somewhat like reducing the type of wavelength, or yes, the distance of wavelength in order to observe detailed structures of the cell. Okay, so the shorter the wavelength, the more ability you'll have to separate small objects. Developments in microscopy. Advancements over time, improved technologies have enabled greater detail in observing cells and their internal structures. Innovations have deepened our understanding of cell functions and enhanced scientific collaboration. In the electron microscopy, we utilize electrons instead of light, and this will offer us a much higher resolution due to electrons' shorter wavelengths. Key features resolve details as small as 5 nanometers, critical for studying cell ultrastructures, such as organelles, membranes. There are some limitations, and the limitation that we can find here is that biological material must be dead due to the vacuum requirement. The electron microscopy types can be classified as transmission electron microscopy, that is abbreviated as STEM. The electron beam passes through thin sections of biological material. Structures stained with heavy metal ions appear dark in the image. Best for observing internal details of cell organelles. Scanning electron microscopy, that is summarized as STEM. Electron beam scans specimen surface, reflected emitted electrons generate a 3D image. Ideal for studying the surface details of specimen. And how it works, the electron beam will be generated by an electron gun. Focus using electromagnets instead of glass lenses. The image view on a fluorescent screen or recorded on a photographic plate. In this case, you can observe, for example, here we have the transmission electron microscope, and it's scanning an electron micrograph of red blood cells. So it's quite crucial to know that the diameter in this case for these red blood cells, also known as erythrocytes, is going to be 5.7 micrometers. And the image was taken from an electron microscope, is in black and white, so we have all over this image for you to see it. Okay, we have the cryogenic electron microscopy, also known as cryo-EM. Okay, so we have the cryogenic electron microscopy, also known as cryo-EM. What is cryo-EM? That is a cryo-etched electron microscopy technique that uses large frozen biomolecules to create high-resolution 3D images. So it will operate at cryogenic temperatures, allowing the visualization of individual atoms within proteins. So the key applications can be, for example, that it will reveal protein structures and functions, that will identify protein malfunctions in diseases, and it will facilitate its drug targeting by understanding molecular interactions. Advantages over traditional methods. Okay, one of the main advantages is that it is compatible with vacuum. It works with delicate biological molecules without destroying them. Also requires lens intense electron beams, preserving the water around samples. Also, it will eliminate reliance on X-ray crystallography, which require crystallization of molecules. In 2017, the Nobel Prize in Chemistry awarded to Jacques Dubochet or Joaquin in Quackintran with Richard Henderson for the development of cryo. We have also the freeze fracture technique. And what is freeze fracture technique? It is a study method to analyze biological structures just by freezing tissues instantly using liquid nitrogen to a temperature of minus 196 Celsius degrees. So tissues will be fractured in vacuum and exposing internal surfaces will have the revelation of fine details. Okay, so the process is basically that we'll freeze the biological material. It's frozen solid to preserve its shape. Solid tissue is broken, exposing internal structures. And the etching, which is the ice, allowed to evaporate slightly, enhancing surface detail. Carbon replica that will be like a mask of the surface that is made would be strengthened with heavy metals such as, for example, gold. And the electron microscopy, which is a replica that is examined to reveal these detailed cell structures. So there are some key insights, such as, for example, consistent images with transmission electron microscopy confirm the accuracy of cell structure observation. And freeze fracture provides high detailed views of internal membranes and other organelles. As you can observe in this picture, the electron micrographs from the inception and freeze etched materials showing the nucleus of a liver cell. Okay, so we can observe the nuclear membrane, that is a double membrane. We also can observe the nuclear membrane with its pore for the RNA to pass through, and the cytoplasm with mitochondria. Also, we have the fluorescence microscopy. It also uses fluorine. Okay, it will use the fluorine atoms with fluorescent dyes absorbing light at one wavelength and emitted at a longer wavelength. So these dyes can bind specifically to target molecules, for example, the oxyribonucleic acids, making them visible under a fluorescent microscope. How it works? So it will work using illumination when light passes through two filters. And the filter one will allow light to excite the fluorescent dye. The filter two will block the excitation light and allows emitter fluorescent light to pass. And the visualization is that dye objects will appear in bright colors against a dark background, as you can observe in this picture. So, 1, 2, 3. There are some applications such as the immunofluorescence. And in this application, antibodies labeled with fluorescent dyes bind to specific biomolecules that can be used in cancer diagnosis, such as identifying infected or affected cells in tissue samples. Cell studies revealing the distribution of DNA, proteins, or cell membranes, and muscle contraction mechanisms such as understanding cellular processes. Examples of images can be seen here. For example, nuclei that is in blue, cytoplasm that is in red, and the damaged DNA that will be colored by green. So immunofluorescence of cancer cells with nuclei in blue, cytoplasm in red, and damaged DNA in green. Now we're going to talk about the common cell structures. Okay? And with the common cell structures, we can write or we can list 3 types of organelles. Cell parts. The first one is the nucleus. And the nucleus is going to be the center, it's going to be like the brain of the cell. It will command all the cell functions. Also, it will serve as storage for the genetic material. It's going to be the DNA storage. And it's the one that controls all the cell function. Okay. And then we'll have the cytosol or cytoplasm. And the cytosol and cytoplasm is going to be like the substance inside the cell that is mainly and more than 70% composed by water. Even more, right? What? Okay, so then we will have the membrane. The membrane will be like the entry, so it will be the entry and the exit of substances. The entry, exit of substances. At the same time, it will delimit the cell from the environment. The cytoplasm and the nucleus also will have some functions. Okay, every single organelle will accomplish with some functions. So as we said, the nucleus controls all the cell's functions. It will be like the brain of the cell. But the cytosol is going to be the substance that will be will be in a regulated pH level in order to maintain some reactions, some synthesis reactions inside the cell. So there are going to be some enzymes, there are types of proteins, reactions that will be using the cytosol or cytoplasm. That is one of the most important substance, I would say, inside the cell. Okay, and the membrane is going to allow the entry or exit of some substances using different types of transports. That is the active transport and the passive transport. Okay. Okay. So there is a vast diversification of functions in each one of these. Okay. So now let's talk about the animal, and the plant cell. What is the distinction between these two? So really important to know that the animal cells can have vacuoles, but they differ from those ones that are in plants. Is that the plant cell will have a permanent vacuole. A permanent vacuole. And this permanent vacuole will store nutrients, but at the same time, it will give structure to the plants. Okay. And okay, comparing other structure, we can talk about the chloroplasts. The chloroplasts that are like it's somewhat like an energy factory for photosynthetic process, is going to participate in the photosynthesis for cells. Okay, animal cells do not have chloroplasts, so by the way, they cannot have photosynthesis. Okay, so there is a concept that I would like to approach today. That concept is going to be called autotrophs. So autotrophs are the organisms that can produce their own food, and heterotrophs, such as the animals, terms. Organisms that cannot produce their own food, but they depend on different orders that are the primary energy sources of them. And that's classified in a food web pyramid. They're going to be consumers, and these will be producers. But there are some organelles that will determine that, such as the chloroplasts or the permanent one. Okay, so other distinction that I would like to make today is the difference between eukaryotic or eukaryote and prokaryote. Okay, so eukaryotic cells, they're the ones that have nucleus. It will have nucleus, so there is going to be a nucleus. But prokaryotic cells will lack nucleus. Okay, they will not have nucleus. Okay. So the eukaryotic means means new, real. I would say real new nucleus. And pro means before nucleus. Okay. Examples of organisms with eukaryotic cells can be plants, can be animals, can be fungi and prototypes. And for prokaryotic, it can be, for example, prototypes. And for prokaryotic cells can be bacteria. Okay, let's talk about the differences between prokaryotes and eukaryotes. So the prokaryotes are going to be smaller in size compared to that one that we find in the eukaryotic cells. We're going to have a nucleus, which is the storage of the DNA information or genetic or hereditary material. And we're going to have this in a one single loop structure. So in the eukaryotes, we're going to have the DNA or hereditary material inside the nucleus. The other difference is going to be the number of organelles that the prokaryotic cells have. In this case, we don't have several organelles as those ones that appear in the eukaryotic cells. So there are going to be some differences. For example, one of them is going to be the endoplasmic reticulum, where some proteins will be synthesized, and these organelles will be divided into subdivisions, which are the rough endoplasmic reticulum and smooth endoplasmic reticulum. Okay. Okay, so basically in size, we can say that eukaryotic cells are going to be greater. The number of organelles also in the nucleus, the prokaryotic cells are going to have a real nucleus as this happens in the eukaryotic cells. And there are going to be some structures that they will be sharing, or there will be some similarities, such as, for example, the cell membrane. A cell membrane that what the test cell in the internal organelles inside of it will be present in both structures. So, for example, the ribosomes. Okay, what else? The cell wall, and at the same time the cytosol. I didn't write the cytosol or the cytoplasm. It is a substance. It is a substance that that regulates and allows the existence of the other organisms. Okay, so I can tell you that the nucleus. Okay, there is a theory that states that is the endosymbiotic theory that states that the eukaryotic cells came from prokaryotes. And I'm just going to mention just one of the examples, such as the beginning of the nucleus. The nucleus, according to some theories, it came from folding the cell membrane And this process took place in a prokaryotic cell, and that was like the creation of the nucleus. And okay, so there are other theories. We're not going to talk about that today. Maybe in another class. And maybe, for example, the mitochondria. And that is another theory that talks about the mitochondrial creation, where we say that this mitochondrion was part of another cell that was consumed by the cell, and it was included inside of its cytosol in order to perform energy functions. Okay, now we're going to see some important concepts, organelles with their size and structure. The nucleus is the largest organelle that exists in the cell, and it goes from 10 to 20 micrometers. Okay. And it will have some pores, and these pores would be minus 30 nanometers. Okay, then we have the chromosomes, and the chromosomes will come from the nucleus. The chromosomes are going to be in two presentations in form of chromosomes and in form of chromatin. And they will be packed by some proteins that are called histones. Histones will be packing the chromosomes. These histones are proteins. They will be packing chromosomes in order to organize this structure or condense it, and we can obtain from this something that we know as the DNA. Okay. Okay. It's quite crucial to know that the nucleus is different to the nucleus, because this is known as a sub-organelle, so it doesn't reach out the category of an organelle as the nucleus, because it's going to be smaller. Its functions are going to be like sub-classified because they have a lot to do in the creation of the ribosomes. Okay. So we can say that the nucleus is going to be inside the nucleus. Inside. This is a number three. Entry, sorry. Okay, so then we're going to have the cytoplasm. And the cytoplasm is also known as the cytosol. We have said this tons of times, and that is a substance that will allow the organelles to have their functions and to keep their pH level in a healthy environment and functional environment. We have the metabolites. There are those substances that participate in metabolic functions. The plasma membrane, that is going to be a layer that is related to the input and the output of material. Right? These are concepts, but some of them are organelles. Okay? This is going to be a concept. For example, this concept, this is a suborganelle, this is an older concept. Okay? It doesn't mean that they all are concepts. Okay, so for example, this is an older concept. It's not an envelope. Okay? And organelles, which are the small parts that we have in the cell, like the nucleus. Okay. So the plasma membrane is that the layer that will allow the income, the, 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 the acid, and also the income of some substances by some protein channels. The organelles are going to be structures that will be inside the cell, like the nucleus, like the mitochondria, like the ribosomes, okay, like the endoplasmic reticulum, which is the endoplasmic reticulum in two divisions. We have it here. Next, we're going to talk about it. Okay, the nuclear envelope is going to be a structure that is going to be protecting the nucleus. Okay. The nuclear pore is related to some small holes that will allow the pass of genetic material, such as the RNA or messenger RNA. And this material will carry out information to the cytosol, and there is some genetic processes will take place, such as the translation, the replication that is taken in the nucleus, but also the synthesis of proteins. Okay. Okay, so then we have another one that is going to be the mitochondria. Mitochondria is going to be the energy factory of the cell, because this is going to be like a source of energy that will convert sugar into energy in order to perform all the cell functions. The mitochondria is going to be the powerhouse of the cell. Okay, and it would be from 0.5 to 1.5 micrometers wide, and from 3.0 to 10.0 micrometers long. The mitochondria is going to perform an important function that is related to the energy production by the production of ATP. And that's going to be related to a molecule that is called the adenosine triphosphate. Adenosine triphosphate will be highly demanded in certain types of cells. So the number of mitochondria is going to be related to the type of cell. For example, in muscle fibers, where the cell energy demand is higher, we're going to have a greater amount of mitochondria. But other important function that it performs is a metabolic function. Because, for example, in liver cells, as the liver must respond to several demands of metabolites and metabolic functions, the metabolites that are the substances that are including this function will be using several mitochondria. Okay, so other type of organelle is called ribosomes. Ribosomes are structures that will be 25 nanometers. They will be formed by proteins and RNA. Their location is in the free cytoplasm, or in some cases you can find them attached to the endoplasmic reticulum, especially to the rough endoplasmic reticulum. Ribosomes are going to be formed by two subunits. They will be formed by a small subunit and by a large subunit, okay? Small and large subunit. It's really important to know that the location of ribosomes is closely related to their function, because you can find them in mitochondria and also in the chloroplasts. In the 70s, they were found in the prokaryotic cells or prokaryotes. They were found in the mitochondria and also in chloroplasts. But in the 80s, there was a discovery that allowed us to identify ribosomes in eukaryotic cells. It's important to know that the ribosomes are crucial in the creation of proteins, which are the units that will allow the functionality of life.
Corte.
Functions of ribosomes. Ribosomes are the workshops where proteins are synthesized in cells, as we saw before. Examples of structural proteins can be the collagen, which is a vital tissue for strengthening. The enzymes, there are other types of proteins, are key to metabolic reactions, acting as biological catalysts that accelerate chemical processes. Ribosomal activity is essential for building and maintaining body structures and facilitating its biochemical functions. The endoplasmic reticulum. The endoplasmic reticulum that is represented with ER is going to be an interconnected network. It is made up of folded membranes, as you can see here in this picture, forming sheets, tubes, and sacs, all connected to the nuclear envelope. The cytoplasmic presence is abundant in metabolically active cells. So, especially those involved in protein or lipid production. The types of endoplasmic reticulums are two. We have the protein factories, ribosomes are attached to its surface, responsible for protein synthesis and modification. And we have the endoplasmic reticulum that is small. The small endoplasmic reticulum will not have ribosomes. It focuses on lipid synthesis and detoxification processes. It's important to consider that the endoplasmic reticulum is central to protein production, lipid metabolism, and cellular detox. Each type specializing in different essential cellular tasks. The ER is central to protein synthesis, lipid metabolism, and cellular detox. Each one of them specializing in different cellular tasks, right? So I'd like to talk about structure of the endoplasmic reticulum. The endoplasmic reticulum is an interconnected network that consists of folded membranes that will give origin to tubes, sacs, or sheets. We have two different types of endoplasmic reticulums. We have the smooth endoplasmic reticulum, okay, and we have the rough endoplasmic reticulum. Smooth endoplasmic reticulum will not have ribosomes, and it will perform the function of lipid synthesis, and at the same time, it will perform metabolic functions. The rough endoplasmic reticulum, opposed to the smooth endoplasmic reticulum, will have ribosomes and will perform protein synthesis and modification. So this is one of the main differences that we find between rough endoplasmic reticulum, because whereas we have a modification here, we're going to have the detoxification in this older one. So it's crucial to comprehend these two differences between these two subparts of the endoplasmic reticulum. The Golgi apparatus consists of a stack-like collection of flattened membranous sacs. One side of stack of membranes
is formed by the fusion of membranes of vesicles from the cell and rel. At the opposite side of stacks, vesicles are formed from swellings at the margins that again become pinched off. Now we're going to continue with lysosomes. Lysosomes are small spherical vesicles that are given in the REP, which is a rough endoplasmic reticulum. Okay, so generally speaking, they are produced by the Golgi apparatus or by the REP, okay, specifically speaking. Lysosomes are going to be formed by some hydrolytic enzymes, and the hydrolytic enzymes are called digestive enzymes. They are going to break down food vacuoles, and they will mix with them in order to obtain all those debris. Lysosomes play a fundamental role in the breaking down of food vacuoles. For example, bacteria that are harmful invaders to the body are going to be detected by macrophages, and macrophages are a type of white blood cells. These bacteria then will be enclosed by food vacuoles, and finally the lysosomes are going to take those food vacuoles in order to digest and process these wastes and debris. Foreign matter or food particles within vacuoles are broken down once they have fused with the lysosomes, and these products will be released into the cytoplasm. Autolysis is a process by which the lysosomes participate, performing a special role in the self-destruction of organelles in the cell. There is another cell process that is crucial here, and this process is called apoptosis. And the apoptosis is a cell program death. It's a programmation in the death of the cell that is taken by the cell in and of itself. And this happens when there is a detection of an infection or when there is a mutation. So it's the most viable way is basically to destroy the internal organelles of the cell, and this happens in organisms, obviously. So lysosomes play important roles here when we talk about this self-digestion. Okay, now we continue with plasma membrane. The plasma membrane is going to be like a barrier that will be formed by a layer that is going to be called a lipid bilayer. And there are going to be some proteins that will identify the substances that go inside the cell and those ones that are secreted or expelled out of the cell, okay? And we're going to have two types of transport here. So we'll have the active transport and the passive transport. The plasma membrane also plays an important role in protecting the cell and delimiting it. Also, it's going to protect the internal substance that is called the cytosol or cytoplasm, okay? There are going to be like some proteins here. There are going to be like some channels to allow the pass of some ions such as potassium ions, okay, or calcium ions. On the other side, it is important to understand that the plasma membrane is another component that helps other cells to recognize this cell. Okay? Due to the plasma membrane, this cell is going to be easily recognized by other cells. is okay. Its size is going to be 7 nanometers. Okay, it's an extremely thick structure. It's an extremely thick structure. The cytoskeleton. The cytoskeleton is a network of fibers extending throughout the cell. The cytoskeleton is going to be participating in structural and also in functional activities inside the cytoplasm in order to have everything working together. The cytoplasm is the substance, as we call it before, the cytosol, and that's going to be formed by other substructures, okay, that are going to be called the microfilaments, and also the microtubules. Okay, the microtubules are going to be formed by some globular proteins, and a globular protein that is called tubulin. So tubulin is going to be forming the microtubules in the cytoskeleton, and it will help organizing every single structural activity that is performed in the cell. Okay. It also has other motor functions. Also, we have other type of substructures that are called microfilaments. The microfilaments are going to be carrying with them some substances in order to make the cell work. Okay, but as we said, microtubules are the thickest structure in the cytoskeleton. They're going to be more or less 25 nanometers. They can be broken down or they can be built up according to the cell's necessities. Also, we have the other substructures that are called the microfilaments. According to the functions that we have with the microfilaments, we can say that microfilaments, these ones, are going to play an important role in cleavage, furrow of cell formation, especially in cell division. Also, they can maintain the cell structure and they also can shape it. And they play a part in cell motility, being crucial, especially in muscle contraction. So here is where we can say that we have finally ended up at least this list. Okay, so we've been talking about some concepts, we've been talking about some organelles in order to understand the cell growth. Okay, now let's talk about processes in unicellular organisms. So the next ones are going to be processes of life. There are 8. The first one is going to be called homeostasis. And homeostasis is related to the maintenance of a constant temperature according to some external conditions and internal conditions. So it's a result of the interaction between these two systems, between the organism that is considered in a certain way as a system, but also between the environment in it. So in response, we will have a regulation that can be called also a thermal regulation that is called the homeostasis, the homeostatic process. The next one is going to be called the metabolism. And the metabolism is a complex network of interdependent and interacting chemical reactions occurring in living organisms. We also have the nutrition, that is basically the process by which organisms take in and or make use of food nutrients in order to gain energy. Then we have the movement. A movement is related to motility. To motion is related to the ability that an organism has to move from one place to another in order to accomplish with other functions. The excretion is related to elimination of wastes that are given due to the previous process that we approached before, that is called nutrition. We also have the growth, and growth is going to be related to the increase in size, mass, and or number of cells. So every single organism will have the ability to grow, and this would happen because there is a cell process that is called the meiosis. And how can it happen? Obviously, with some with other process that is called the cell division. Okay. The other one is going to be called a response to stimuli, that is also known as sensitivity. So organisms are going to be able to respond to the different stimuli that exist surrounding them. Okay, so they are going to be able to create a response to certain conditions. And finally, the reproduction. And the reproduction can be taken according to two types of reproduction, which are the sexual reproduction that depends on two different individuals. Okay, and obviously they got to belong to the same species, and there is another one that only depends on one single individual. That is called the asexual reproduction. And with the reproduction, they are going to guarantee the formation of new individuals.